Hey IB Environmental students, here's your first IA Support podcast on the DCP and DEC sections of the lab. Here are the expectations and tips, and those are really the only things you should be writing down in your lab part of your spiral as we go through this. You don't need to write down every single example. Pause, though, to read the examples in depth. All right, superheroes, let's go. Keep your spirits high. Okay. So when we're talking about these internal assessments, they're all based on 3 different rubrics, and we're only talking about 2 of the rubrics, and here they are. The first rubric is called the DCP section. It is got 3 aspects, those are the columns, and each of them are added 2 points, for a total of 6 points. The second rubric is, the discussion, evaluation, and conclusion, the DEC rubric. That is also 3 aspects, each of them out of 2 points. Now remember, this is really tough rubrics, so you have a couple different things at your arsenal to help you through understanding these rubrics. Those are that how to write a good ESS lab report packet that you got a paper copy of. There's lots of good things on Blackboard, also on the internal assessment tab. And you have that not starting with a blank page IA template that you're always supposed to start with. So when we're scoring these, we have to understand that these have really high expectations. And because of that, when you're trying to get your class grade, which goes in the grade book, I use a conversion scale. This conversion scale will be available on Blackboard, and you'll notice that it is specific to different types of IAs, depending on if they're out of 6 points, 12 points, or 18 points. But always, about half the points is a 75%. Now that might seem scary, and I think it is a little scary, but you have to understand that these high expectations will help you be the best lab writer you can be. And you guys can be great lab writers. You are superstars or superheroes. So let's keep a positive outlook and continue on looking at what those sections are for the DCP and DEC. Remember, don't write down the specific examples, only write down the tips and expectations. Pause to read the examples, though. All right, let's begin. The first part is writing hypotheses and aims. Here are some examples. This is an example that is way too vague. It doesn't give me enough information. And also, there's no research question or aim. This one is actually the opposite problem. It does give me a research question, but it is blah, 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 very long. And this lengthiness actually doesn't make a lot of sense. So be careful. You're welcome to write a lot, but make sure it makes sense. And this hypothesis is at least a little bit more clear, but it is still a little bit confusing. Here are some more examples. This one has been broken down into an intro, question, hypothesis, and rationale. Do notice that anytime you have an organism, you need to actually write its scientific name down. But having this rationale section allows for us to think about why we picked the answer to the research question we have, because a hypothesis is just an answer to an experimental question, not just a prediction. So we always want to back it up with a rationale. Usually that rationale is within this section of hypothesis and aim. Here's another strong example that you should read over by pausing. Do notice that this hypothesis is not as strong as the rest of this because it is a little overly general. I should have a little bit more information in there about what I'm comparing plastic to. Alrighty, next section. Now we're starting the parts of the DCP and the DEC. You'll see in orange that we're always going to be referring back to the rubric, what aspect we're talking about. The first aspect 1 is all about the raw data, the data that's not manipulated. And you'll see from this student work example that this is a big no-no. Why? Because this is ugly. Way awkward, turtle. We never split a table across 2 pages. Look at a split turtle. Never split a table. Okay, here's another example of a split table. This is a big no-no, but you'll see that at least their tables have numbers, and you always want to do that so you can refer back to tables when you're writing about them. And your tables should always have clear titles that are full sentences that make sense. This one's not fully grammatically correct and it's a little confusing. Here are some other examples of tables. This one is got at least a clearer title. The headings need a little bit help, but definitely the body of the table is a big no-no, awkward turtle. We never put the units in the body because this is weird. Look how much this is repeating itself, and it's weird that it's in different wording here. So we always put units in the headings. And often, if depending on the lab, we're also going to want to put uncertainties in the headings as well. Next section, aspect 2 of the DCP, all about data processing, which is usually in an overview, and the sample calculations. Here is what my expectations are down below. Here's an example. This is straight off of Turnitin.com, so it's a real student example, and here's my Turnitin.com feedback to this person. I can't see in their overview section how and why they decided to average their data. I also should, somewhere down below, will have comments that I don't see actual equations for what these samples are based on. And I'm also missing sample calculations for averages, and that's a big no-no. So make sure you're very clear. Next will be a couple other examples. You pause and see what's wrong with them and check to see if you agree with me. Here's another one. This one has broken it down into table headings. They have no equation for mark and recapture presented here, so that's a big no-no. But they at least have a little bit more information about how they're averaging. Here's another example. This time I have a clear equation, but now I have the calculations weirdly embedded into a table, and that's not very clear at all. This is a bit confusing. So I want to have my sample calculations out separate and clearly labeled. Now on to presentation, the third aspect of the DCP rubric. Here are the rules for how we present process data. We carefully need to pick some way to clearly and meaningfully present the data, usually tables or graphs. If we're not sure what to use, we go to the IA information or resources on Blackboard. But really important, we never, ever, ever are going to present raw data, and that's what's wrong with this. This person has not just included averages, the process data, they've included every single raw data point, and there's no way you can write a clear trend or pattern based on this. Here are some more examples of ways to present process data. For some labs, maybe a pie chart's appropriate, and if so, we always have to have a clear key and want to clearly label stuff. We should always title it with a figure number so we can refer back to it later in the conclusion. Here's a good example of a bar graph. Clear units are apparent. We have a title, but we need to have it labeled figure number something. But what's missing here is error bars. Error bars help us see how reliable our data is and how closely they are to each other. If you need help with error bars, go to the IA resources on Blackboard, but because they're missing, this person wouldn't have gotten full points, even though it's a nice figure. Lastly, don't forget that not all IAs need to have graphs for their presentation form. Sometimes they're just tables that are really clear. So here's an example of that. This one could be a little clearer, and I think we also have to always be very careful of significant figures. My comments here were there could have been a single decimal place after the 20, and that way we could get some extra information. Our next section is about discussing and reviewing and analyzing results. Pause so you can read this and note my comments. But overall, this section, which is the first section of the conclusion rubric, this needs to have specific patterns and trends just like we practiced in class. And those specific patterns and trends should have actual data and numbers involved in them. And then you should be referencing that process presentation of data. Like figure 1 says blank. We also want to note any error by citing specific evidence of anomalies, outliers, error bars, or standard deviation. Remember, we're humble scientists. We know our experiments aren't perfect. Lastly, we want to make this broad implication connection, thinking about what kind of consequences our results might have, connecting back to our original aim. So read over this example and think about my comments I have over here. Our next section is about evaluation of limitations and suggestions for improvements, the second section of the conclusion. I highly recommend this format that's a table because it'll help you avoid mistakes. And here are information about acceptable versus unacceptable things for this section. Pause to read them over. You'll notice that the unacceptable examples are very short and the acceptable examples are long. Just because it's long doesn't make it good. What makes it good is that it has a significance of why the limitation is a problem. The unacceptable examples are very vague and don't tell me squat. So for instance, the test tubes weren't clean. Well, why do I care? What would that do to my results? Do they make them unreliable, unpredictable, not precise, inaccurate? Human error. Well, what kind of human error? What's the problem with that? And not only that, I always want to give a very specific improvement for each of my limitations or weaknesses. For our class, we're going to mostly focus on weaknesses or limitations, not strengths of our experiment, because actually IB is more focused on that too. So when we're talking about these significance and improvements, we always want to refer back to these three important guys: accuracy, precision, reliability. Don't write them down in detail in your notes because you always have them available in your How to Write a Good ESS Report packet. Overall, here's the important things to keep track of. Be specific, be detailed, and every limitation needs a significance and improvements. And I really believe strongly in this format. Here's some examples that aren't so strong. Oh, the data collecting was off. Well, why was it off, huh? What's the significance of that? How will you do that better? Oh, we weren't careful. We didn't calculate correctly. Well, what would be calculating correctly? What's the significance of doing that? And how specifically are you going to do that better? Will you do more trials? How was this human error? This is very unclear. This organizes also a scapegoat that is not clear either. So you really need to be very, very careful in this section to be specific and not be over superficial and simplistic. So here's the strong example. Pause to read it over and carefully. Notice they have used this format and it is a full point. They got perfect on this aspect. Our last section is really just the conclusion. And to do a conclusion, we have to restate our original hypothesis, remembering to be humble, because we're going to support or reject that original hypothesis, thinking about error. Now, we never can prove, be wrong, or be right when we're doing this. We're humble. We're just deciding how likely this supports what we originally thought was the answer to our research question or rejects. Now, this person right here in this last sentence is very humble. They say that the percent error is so high that their data might not be reliable. This is awesome. They got full points on this section. But most students are too scared to be humble. You can do it. You can be humble. But in this conclusion, you have to reason using specific data as evidence of why this is the trend. This is going to look a lot like the first aspect of the DEC section, and it might repeat itself a little bit, especially because not only do you write about patterns and trends using specific data points, you're also once again going to connect back to the aim or question. What's the big picture? What does this data mean? And here's another example of using specific data and making sure to be humble, saying we don't support the original hypothesis. And that's okay. Some of the most important scientists in history actually found out that they didn't support their original hypothesis. And that's actually really cool and okay. You guys made it, and you can do this. I know IAs are hard, but you are superheroes. You can always see Blackboard for more help or always ask me for questions. All right, see you guys in class. Bye.